So thank you, Alain, for organizing this symposium and giving me the hard task to end the day after so many beautiful talks. And so I will tell you today about the work of my team here at the Cerveau in the Collège de France, which is interested in understanding the mechanisms that control the formation of neuronal networks in the mammalian brain. And so the brain, as many of you know, is composed of many different types of neurons, not only in terms of their morphology, as illustrated here, but also in terms of their physiology. And these neurons need to be connected in a precise manner, sometimes over long ranges, to form a functional brain. And any defects in this process will lead to neurodevelopmental disorders such as autism or schizophrenia. The development of these functional networks involved several steps, and our team is interested in the last ones, which is once a given neuron has sent its axon towards the target area, how does it choose its partner, and how does it decide where on this partner it's going to form its connection or synapse? On top of this recognition process, there are also other processes such as refinement, in particular through activity-dependent synapse elimination. And finally, these very specific neuronal networks need to be actively maintained into adulthood so that the brain is functional, hopefully, for many years. A nice example of this very specific connectivity is found in the cerebellum, more particularly on Purkinje cells, which receive, amongst others, two excitatory inputs, two types of glutamatergic synapses, one from parallel fibers that arise from granule cells. And each Purkinje cell will receive about 150,000 of those synapses, which bring multimodal information. Purkinje cells also receive glutamatergic synapses from climbing fibers that are the axons of the inferior olivary neurons. And each Purkinje cell receives one climbing fiber that will make about 300 synapses that will drive a null or non response in Purkinje cells. And these fibers bring topographic information. What is interesting is that these two inputs make their connections on very distinct territories on Purkinje cell dendrites. Parallel fibers make their synapses on spines that are on distal dendrites, whereas climbing fibers make their spines on the proximal or thick dendrites of Purkinje cells. Also, there is an activity-dependent synapse elimination process occurring in Purkinje cells during postnatal development, since at the beginning, Purkinje cells receive 3 to 4 climbing fibers, and then a pruning process leads to this one-to-one relationship. And finally, this connectivity is maintained since it's well known that if you remove parallel fibers, even in adulthood, the climbing fiber will increase its territory. So it's a nice illustration of what I told you before. This is not particular to the cerebellum, since one can find this type of connectivity even in other regions of the brain. So for example, in the somatosensory thalamus, relay neurons will also receive several types of glutamatergic inputs, synapses from the cortex, which bring also multimodal information that will connect those relay neurons on the distal dendrites. And they receive also glutamatergic synapses from the trigeminal nucleus through lemniscal fibers. And here also we have this one-to-one relationship with these synapses being made on proximal dendrites of these neurons. And it's also a strong all or none response that is induced by those lemniscal fibers that brings somatotopic information. And finally, a close example also is found in the dorsal cochlear nucleus, where pyramidal cells also receive two types of glutamatergic inputs. One is also from what is called parallel fibers that are very close to, that arise from neurons that are very close to the granule cells that are found in the cerebellum. And these also bring multimodal information. And they receive also inputs from auditory nerves that are also glutamatergic and bring the tonotopic information. And these two inputs are also made on very different dendritic parts of these neurons. So the question is, how can we instruct the formation of this very specific connectivity on such different neurons in terms of their morphology and function? And amongst the different mechanisms that can control this, one hypothesis that has been postulated several years ago, now 50 years ago, by Sperry is this chemoaffinity hypothesis, which, as Erin suggested earlier, states that for each connection type in the brain, you would have a different combination of protein that would code for this specificity. And so we are interested in understanding whether there is indeed such a molecular code in the brain, in the mammalian brain. So as in our lab, we are using the olivocerebellar system as a model. So the cerebellum is this structure which is in the back of our brain. What is the cerebellum for? I'd like to illustrate what it does with this video. So as you can see, those dancers have to really regulate the rate, force, and rhythm and accuracy of their movements. Otherwise, they would be slapping each other's face. And this is actually the cerebellum that regulates that. And interestingly, it's now increasingly recognized, as illustrated by this recent review, that the cerebellum, in the same way that it would regulate motor coordination through this process, would also regulate the speed, capacity, consistency of cognitive processes. So it's a structure that is getting a lot of increasingly interest. And for us, this is useful because any defect in this network will have a clear behavioral correlate because it will affect motor coordination. So if you have genetic models, you can right away see the defects. But more specifically for us, the advantage of working in the olivocerebellar system is its well-described connectivity and physiology. So the cerebellum is a multi-layered structure, and its surface, the cerebellar cortex, is organized in several layers. And we know the cell types that form these layers. Those are illustrated here on this drawing by Ramon y Cajal, where you can see the beautiful Purkinje cells that are the sole output of the cerebellar cortex, granule cells, interneurons. And we know exactly how these neurons are connected to each other. So, as I said before, Purkinje cells receive two excitatory inputs, one from inferior olive, the other one from granule cells that form the layer underneath the Purkinje cell layer. And they also receive inhibitory inputs from stellate cells and basket cells. Another advantage is that now we have genetic access to each cell type in this network. And this is illustrated here with this slide of the use of bacterial artificial chromosome to generate transgenic mice. And here this illustrates how we can drive GFP expression, specifically either in Purkinje cells, interneurons, or granule cells, depending on the regulatory elements that you have on the specific back. And using this strategy, this back transgenesis, this type of drivers, we can now do many manipulations in the mouse. Neuron-specific optogenetics, if you drive the expression of channel rhodopsin in a given neuron of interest, but also gene expression profiling using the backtrap technology or synaptic protein profiling, as I'm going to illustrate later in my talk. So the question is for us now, using this system, and in particular, the fact that those Purkinje cells receive those two types of glutamatergic input that are very specific, the question is to find whether there is indeed a specific combination of proteins for each one of these synapses, and whether, if this specific combination exists, whether it has a role really in regulating the development of this specific connectivity. So to address this question, we performed a gene expression analysis in the network. The reasoning was the following. If you know all the genes that are expressed by each partner in the network, and in particular by the two afferents, excitatory afferents on Purkinje cells, you can compare those profiles and extract from these the differentially expressed genes that are specific to each afferent. And in this list, you should be able to find the genes that code for membrane or secreted proteins. And if you compare those with the gene expression profiles of the target neuron, then you should find ligand receptor couples that are specific to each one of those synapses. To do this, we use the backtrap strategy, which uses the expression of tagged ribosomal proteins in specific neurons in transgenic mice. And this had already been done by the Heinz lab to profile Purkinje cells and granule cells. So we only had to generate another specific mouse for the inferior olivary neurons, the third partner in the network, which is what we did. And then we used those mice to profile the inferior olivary neurons and perform our gene expression profile comparison. So we used a very high threshold when we performed our comparison to really extract the genes that are really differentially expressed between the two afferents. And when we did that, we identified about 500 genes for each Purkinje cell input. And and we looked at the genes that were at the membrane or secreted. And what was really interesting was that we found for each afferent a different combination of proteins of the C1q complement family. So what is this protein family? It contains the classical component of the classical molecules of the complement family that are known for the innate immunity, such as C1qa, but it also has many other members that are classified in different subfamilies, which all contain this globular domain C1q. And they all have the ability to form multimers, and that's how they become functional, basically by forming these multimeric structures. As I said, we have different subfamilies, and those molecules have been increasingly interesting in the field of synaptogenesis, because we know for some of them that they play a role in the development of synapses. So one of the examples is the cerebellin subfamily, in particular cerebellin 1, that has been shown by two Japanese groups, the Yuzaki group and Mishina group, to be the ligand for the GluR delta 2 receptor and to promote synapse formation between parallel fibers and Purkinje cell synapses through this interaction. So the cerebellin seems to be a promoter of synaptogenesis. The other proteins of these families that have been shown to play a role in synaptogenesis, in particular by Ben Barre's lab, are the classical component of the complement family. And they have been shown to actually regulate synapse elimination during development through an interaction actually with astrocytes and microglia. And the last subfamily that I'll be telling you more about today is the C1q-like subfamily that has been little studied. A recent study from the Sudaf lab showed that this C1q-like family seem to promote synapse elimination in cultured hippocampal neurons. So what we see is that from our data, we identified a different combination of these proteins of this family at each afferent of Purkinje cells, and we've checked these data by in situ hybridization for some of them, and we've identified, and this had already been published, that there's a high expression of cerebellin 1 specifically in granule cells, whereas climbing fibers express high level of C1q-like 1 in addition to cerebellin 4, which has been little studied up to now. So, is this combination instructing the specificity of our connectivity on Purkinje cell? Before getting to this, I have to make a little pause and to go back a few years so that the future can make sense. What we did a few years ago when I was in Nat Heinz's lab was to develop a strategy which we call synapse protein profiling that enables one to purify given a specific synapse from a specific type of neuron from the mouse brain and identify its protein composition using mass spectrometry. And we did that for the parallel fiber Purkinje cell synapse. We identified about 60 different proteins in our analysis, and these proteins encompassed all the categories that are classically found in studies of postsynaptic densities by mass spectrometry. And we were, of course, particularly interested in the receptors that are found at this synapse. And here we found the presence of this receptor, the brain angiogenesis inhibitor 3. This receptor is interesting because of several characteristics. First, it's part of the little studied subgroup of G protein-coupled receptors, the adhesion GPCRs, which in addition to their 7 transmembrane domain, contain a long extracellular domain that has basically recognition modules, and in this case is thrombospondin type 1 repeats domain. The BI3 receptor is highly expressed during development in many regions of the brain, including the cortex, hippocampus, but also in the cerebellum, as you will see. And it's been associated by several studies with psychiatric disorders. Despite all this, the role of the brain angiogenesis inhibitor 3 receptor during development was unknown until recently. So during the development of the cerebellum occurs many morphogenetic steps. And in particular, Purkinje cells have to go through a very intense remodeling activity to generate their mature dendrite, the adult dendritic architecture that you've seen before, that is very elaborated with a lot of branches and that are also in 2D, which is really interesting. On top of this intense generation of branches on Purkinje cells occurs also synaptogenesis from the granule cells that migrate and at the same time grow their parallel fibers that will contact the Purkinje cell dendrites. And at the same time, we have the synapse elimination process that I told you about before, where we have the elimination of supernumerary climbing fiber to reach the adult stage where we have 1 climbing fiber per Purkinje cell. BEI3, by in situ hybridization, can be detected in Purkinje cells throughout this process. So we wondered what role it could play during the development of Purkinje cells. And first, we tested its role in regulating dendritogenesis. So this study has been published recently, so I won't go into the details, but I want to illustrate you how the adhesion GPCR BEI3 regulates neuronal architecture, in particular by describing the experiments that we done using RNA interference against BEI3 in the mouse cerebellum during development. So if you first we knocked down a BEI3 at P4 in mice and looked a few days after to see what was the effect of knocking down this receptor on the acquisition of the dendritic architecture of Purkinje cells. And as you can see here, whereas a control small hairpin RNA will have no effect on the growth, the direction of growth of the primary dendrite and its branching when you compare to non-infected cells. If you knock down BEI3, there's a profound effect with dendrites that grow really not the right way and are not properly differentiated. And if you look later, at later stages, this defect can still be visible, as you can see here with this primary dendrite that really doesn't grow toward the proper direction compared to controls. So this and other experiment with transgenic mice really showed us that BEI3 is important for the development of the proper architectures of neurons. But I told you before, we found BEI3 at the parallel fiber Purkinje cell postsynaptic density, and in cultured hippocampal neurons, we can detect very high levels of BEI3 at spines. So we wondered what was the role of BEI3 during synaptogenesis. And so we looked again at higher magnification at the neurons that were transduced with lentiviruses driving either an SH against BI3 or a control SH. And as you can see, already we can find defects in the spines, in their shape and in their density. But because quantification of these spines is difficult in vivo, we turn to cerebellar mixed cultures, where we could analyze, after staining with focal binding, the dendrites and spines on Purkinje cells after knocking down BI3 or in control situation. And as you can see, knocking down BI3 significantly reduces the density, but also the total number of spines in Purkinje cells. And this effect is principally principally affecting the mature spines. So the addition GPCR BI3 regulates spinogenesis. What about synaptogenesis? So what is convenient is that in Purkinje cells, in adult Purkinje cells, one can stain specifically the synapses from climbing fibers with a VGLUT2 marker, and the ones from granule cells, as I will show you later, with another presynaptic marker, VGLUT1. So first we stain for the climbing fiber using an anti-VGLUT2 antibody. And what you can see here is that in normal conditions, the climbing fiber will climb up the Purkinje cell dendritic tree up to a certain level. Whereas in the situation where BI3 is knocked down, this doesn't seem to be the case. And quantification indeed confirmed that there was a deficit in the translocation of the climbing fiber on the Purkinje cell dendrite when we knocked down BI3. We then also looked at the puncta for VGLUT2 in the situation where we knocked down BI3 and in the control situation. And our morphological analysis showed that indeed we had a significant reduction in the volume of the VGLUT2 clusters when we knocked down BI3. And electrophysiology experiments showed that indeed the transmission between the climbing fiber and the Purkinje cells was affected when BI3 was knocked down. What happens to the parallel fiber Purkinje cell synapses? Electrophysiology again showed a profound defect in the transmission from parallel fibers to Purkinje cells when BI3 was knocked down. And we turn to cerebellar mixed culture to quantify this at the morphological level. And as you can see, indeed, the volume of the VGLUT1 clusters is reduced, and the volume of contacts is significantly reduced when we stain for VGLUT1. But when spines are there, they are contacted. So there's no significant difference in the numbers of contacted spines. So that means that each spine that is there is contacted, but there is a deficit in the maturation of this synapse. So what I've shown you is that BI3 can regulate synaptogenesis and the maturation of synapses for both afferents on Purkinje cells. So here we go back to the results of the complement family. I showed you before, we have a different combination of proteins of this family for each afferent. What's the relationship with the BI3? Well, cerebellins, as I said, are important for synapse formation and stability through their interaction with GluR delta 2. Interestingly, the study of the pseudoflam showed that C1q-like 1 is actually a ligand for BI3. So with this in mind, what one can postulate is that this specific production of C1q-like 1 by climbing fibers is actually what regulates the maintenance, the formation and the maintenance of the climbing fiber territory on Purkinje cell, because one can postulate that this is this C1q-like 1 secretion that affects and prevents the parallel fiber from connecting their territory. So this is the hypothesis that we have been testing. And so the prediction would be that if you misexpress C1q-like 1 in the other afferent, because this would prevent even more the stabilization of the parallel fibers on Purkinje cells, this would induce their elimination, you should increase the territory of your climbing fibers. To test this, we used again lentivirus to drive this time not shRNA, but either GFP or the C1q-like 1 protein. And in the control situation, you can see some parallel fibers that are stained in green here. And you can see your climbing fiber that is stained by the anti-VGLUT2 antibody that crosses those territories and climbs up the Purkinje cell dendritic tree. In the situation where we overexpress C1q-like1, what you can see here is that the climbing fiber does not seem to be able to cross this boundary. And we've quantified this and shown that indeed, when you overexpress C1q-like1 in the cerebellum, you have a significant reduction in the extent of the climbing fiber territory on Purkinje cells. So this is actually exactly the reverse of what we predicted. So what does that mean? A simple explanation would be that actually C1q-like1 does not inhibit synapse formation, but actually promotes it. And so if you promote the parallel fiber territory, you will reduce the climbing fiber. We are currently analyzing this, and we have data that indeed suggest this. So if you knock down C1q-like1 in cerebellar mixed cultures that do not contain inferior olivary neurons, you only have granule cells and Purkinje cells, and stain with Cal binding to show the Purkinje cell dendrite and their spines, and VGLUT1 to show the parallel fiber synapses, it seems that indeed we have a reduction in the number of synapses on those Purkinje cells that can be rescued. This is interesting because indeed, transiently, it had been already published that the precursors of granule cells can produce C1q-like1, albeit at a very low level compared to cerebellin 1, which is induced afterwards during the maturation of granule cells, and these are data from our lab. So this is how we're putting this together now. Basically, at early stages, C1q-like1 is secreted not only by climbing fibers, but also by the granule cell precursors, and this would promote synapse formation on the Purkinje cells by the excitatory inputs. At later stages, C1q-like1 is still expressed by climbing fiber, but it's actually the switch in granule cells from secreting C1q-like1 to secreting cerebellin 1 and 3 that kind of removes a brake on the climbing fiber and allows it to translocate and stabilizes its own synapses. So basically what we're seeing here is a picture where we have a different combination of complement proteins, proteins of the C1q complement family in each afferent, and a different combination of receptor postsynaptically that would define each one of these inputs. So finally, what this means is that we're getting at this type of molecular synaptic code using our strategy, and in our gene expression profiling and in our comparative analysis, we have identified other proteins that might be important for this process, and in particular, special note for Sonia, and actually Antoine too, the major difference actually that we found at the functional level between the differentially expressed genes in inferior olivary neurons and those in granule cells is that inferior olivary neurons express a high diversity at high level of proteins that are found in the immune system as well. So we are now interested in looking at why these inferior olivary neurons need so many different proteins of the immune system and whether this has something to do with their connectivity on Purkinje cells. So finally, I'd like to thank the people who did this work, especially the people of my team, Séverine Siguayo, who did a lot of the work on BAI3 and C1q-like1, Alessia and Kertana, who did a lot of work on the gene expression profiling and comparative analysis between inferior olivary neurons and granule cells, Maeva, a technician who contributed to all of the work, and also all the facilities in the CIRM that are essential for this work. Our collaborators, and in particular Philippe Izop and his postdoc Francesca for the work using electrophysiology, as well as Nat and Joe for their help in the backtrap analysis. And these are our funding, and thank you for your attention.